everyone for coming. Today's speaker is Jasper Weinburd. He's an NSF postdoc at Harvey Mudd College, working with Andy Burnoff on pattern formation and dynamical systems. He got his PhD in 2019 from the University of Minnesota, so we're buds from that time. He also is really dedicated to working with undergraduates. He spent the summer working with 2 undergrads on a project on locust modeling as part of the Harvey Mudd College Data Science RU. He has lots of good information, like in general, and I'm really looking forward to this talk about emergent patterns and locust swarms. So feel free to catch up with him in the gather room if you have questions just about life or about the talk in particular. Sorry, Jasper, volunteering you for that.
I'll do what I can.
Yeah, all right, so take it away.
Okay, thanks, Alice. And thank you all for having me to give this talk today. I need to thank one other person who I'm not sure if she made it, but I was originally invited by a postdoc at Cornell who's no longer there. So thanks also to Kate Meyer, who Alice and I both know from graduate school as well. So let's get into it. This up here in the, you all can see my cursor, right? I think I checked that earlier, but great. Thanks, Alice. This is one locust. Basically, they're grasshoppers, right? Maybe you've read about them in the Bible or something like this. And these 2 pictures, one is from Australia and the other is from Africa. And in each of them, we're looking at an aerial view of a swarm of locusts. So this darkened area is where the locusts are in this picture. And again, here, they're streaming through in that way. And these pictures are going to pop up again and again throughout the talk today. One thing I want to highlight the first time you're seeing this is that locusts eat a lot of vegetation. So on the left, behind the swarm, the locusts have essentially decimated this field. And on the right, ahead of the swarm, there's still some nice green, lush vegetation. One other quick comment. We're here in the time of COVID, and although she's very cute and looks like just the most wonderful puppy in the world, Riley's bark sounds like she's a 100-pound hound. So if you hear Riley barking, we'll just move past that and let me know if you can't hear what I'm saying anymore. But she doesn't bark too much. It's just when the mailman comes. Just one of the things we're dealing with these days, right? Okay, so we're all familiar with one plague this year. COVID-19 obviously is the reason I'm not there in Ithaca with you today. But there's actually another plague, a plague of locusts, which has been spreading. It originated in southwestern Asia and has spread into the Horn of Africa. Most recently, it's even making its way into southern Africa. So I pulled a few headlines about just to highlight how these locusts are not just an ancient biblical concern, but they're a real concern today. This you'll see is from January 2020. Starting April 2020, you see a headline including both plagues, some devastating interactions there. This one, a little bit later, August this year, and just from a week and a half ago, a couple weeks ago, what's going on now is that in Pakistan, southwestern Asia, the plague is starting to subside, but all these swarms are threatening southern Africa at the moment. So this is a current real issue that locusts are threatening food security throughout the world today. We're going to talk about how these locusts swarm before I can pose the research area and the mathematical modeling that I'm working on. So I showed you this one picture of a locust here. And the locust on the left is kind of like a locust who is doing a really good job of social distancing, right? This is a solitarious locust, and these locusts will forage on their own, and they'll actually avoid other bugs, other locusts. And that's their typical state. But in the right environmental and conditions, locusts end up crowding together, and they undergo this phase change, epigenetic phase change, which they'll then pass on from generation to generation. And on the right-hand side, we've got gregarious locusts, which are attracted to other locusts. So you get social aggregations and social interactions. So this is kind of Jekyll and Hyde, or social distancing and ignoring social distancing behavior to locusts. So how does this play into the creation of a swarm? Well, let's say we start with 4 or however many, some solitary locusts. And for whatever reason, food in a certain year or time period is very sparse. Well, what happens is the locusts will group up onto that food, and this phase change can take just hours, they'll start aggregating together. Then the physical changes after the behavior change take a bit longer, a couple of generations. So now you've got a bunch of gregarious locusts all sitting on a bush, and typically they hatched at the same time together. So from there, the locusts will start migrating and foraging as a collective group. So hopper bands are is the name of this stage because the locusts haven't developed their wings fully yet. So these juveniles are marching across different landscapes as they migrate to better resources or eat their way across a field. And then, of course, the final stage is what parts of Africa are dealing with right now. And these flying swarms are the most devastating. And as we saw before, they can travel huge distances, even crossing bodies of water like the Red Sea. So we're going to focus today on these hopper bands, which are on the ground. They are marching and hopping. And they're also a key stage in order to interrupt this development of the big swarm. So if your locusts can fly, it's much harder to spray them with pesticides. So they're a key stage in the development of a swarm from a control perspective. So let's zoom in a little bit. I'm going to play this video from BBC Earth just to give you a look inside of a hopper band. And you can see a couple of different things as these. You can see them marching, walking, and then you also see ones that hop. You also see how they're all essentially moving in the same direction. That's one of the well-documented social interactions. And then here at the end of the video, you see they've stopped marching in order to eat. And that, of course, is the biggest concern from an impact on environment and humans perspective. So here are these 2 pictures that I showed you earlier. And this is a quote emphasizing what I was just saying, that at a critical density, locusts will engage in this coordinated or directed collective motion. So as I mentioned before, these are from 2 different species, but it turns out that you can see these different types of formations in each different species. And the different types of formations I think of here is I'm looking at this one, and all the locusts are moving in this direction, perpendicular from the overall structure. So with my background from partial differential equations, nonlinear waves, I look at this and I see a planar front. So that's what I'm going to call this shape here on the right. Or on the left, I'm sorry. On the right, all the locusts are moving parallel to the overall structure. And I call these kind of columnar structures. So these are the 2 emergent patterns that occur in hopper bands that I'm sort of use as 2 ends of one extreme. And over the years, even going back to 1950s or before, one idea you might have, even just looking at these pictures, is that maybe it's the vegetation that's playing a role in determining which of these collective structures occurs here. Right. So this is kind of the big questions for me in this research program. These collective structures, what role do they play as effective methods for the locusts to survive and propagate or migrate? Also, how do the individual level mechanisms cause distinct patterns to form? Is it this food, or is it the differences in how locusts are attracted to each other or interact with each other? And down the line somewhere, my hope is that understanding these better will help in programs of control like pesticide spraying or barrier methods where you basically have a piece of land that you don't plant crops on. The talk today is going to be split into two pieces. And these two pieces focus on modeling the planar front on the left and the columnar structures on the right. So we have a couple different frameworks for each of these situations. And I'm going to talk first about the one on the left. And this work is kind of a complete story and published. The work on the right is in preparation, and in some parts, in terms of some data collection, is actually still ongoing. Oh, and I should play my videos. So these are videos of our two models, a little preview for you. So that's our plan for today. Let me start by giving some quick selective review of agent-based models, and in particular, the flocking type of model, which is, so you can tell where I'm coming from. So this image is an image showing how some focal individual adjusts its orientation depending on its nearby neighbors. This kind of goes with Reynolds' original Boyd's paper. So that's sort of the context and the type of agent-based model that I'm looking at here. I also want to highlight this. This is actually a picture from a modeling and experimental paper about locusts. And it highlights that stop and go type of motion, that jerky motion we saw in the videos. So you can see that locusts are switching between standing and walking, and they're in some kind of laboratory arena here. The last thing, which will come up in the second half of the talk, are these zonal models, or three-zone models of social forces. So the idea here is that nearby locusts are repelled from each other. Intermediate range locusts are track their alignment with other locusts in that range. And at far ranges, locusts are attracted to other neighbors. And this is a sort of, this is documented and used throughout the agent-based model biology literature. And specifically in locusts, it's also been seen in some empirical observations as well. Lastly, there has been some previous work in documenting these different types of patterns that you can see, essentially by changing parameters, internal parameters within the models. And two things stuck out to me and my collaborators when we looked at this, the efforts that have been made in this area before. So one is that the same group of locusts appear to demonstrate differing patterns in differing environments and based on different times of day or things like that. So in varying internal parameters would suggest something like, well, this locust species always forms this pattern, that locust species always forms that. And we're not quite satisfied with that. The other is none of these efforts incorporated food prior to our work. And so we're going to look specifically at that question first, of how does food affect the types of patterns that are forming here. So we're specifically inspired by this planar front image and some field observations of locusts. So I did already say this is going to be a one-dimensional model. So let's take a cross-section here. And let me just flip the picture from the field observations quickly so that it matches the directionality that the locusts are moving in our picture. This data is essentially collected by a field ecologist, Jerome Buell, He's down at University of Adelaide in Australia. He set up a camera and then watched a locust band march underneath and took video and then counted locusts in frames. And the key observation here is this pulse-like structure where you have a steep increase in the front and then an exponential tail in the back. And he quantified its fit to an exponential and the exponential rate and things like that. So from, as I said, my background in nonlinear waves, this is like a big air siren saying, hey, come study this with a traveling wave methodology or model. Okay, so let me quickly introduce our agent-based model. And I'm going to think, okay. So we're looking at a one-dimensional cross-section. We think of this as employing that social alignment and including that implicitly in our model. So all these locusts are moving in the same direction anyways. Let's use a one-dimensional model. And so take n locusts, and they're going to be split into either stationary or moving. There's something to be said about hopping as well. But for simplicity's sake at this point, let's keep it at either they're stopped or they're moving. And each time step, locusts switch between these states with some probability. And then we just iterate this simple algorithm. If they're in the moving state, they move to the right one lattice point. And if they're stationary, they feed locally. We model resources with just a spatially discrete resource field, which decreases near stationary locusts. These probabilities, PSM and PMS, these are what couple the resources with the locust density, or the locusts. And they do so by depending, so if you're a stationary locust and you're going to switch into the moving locust, then your probability depends on the local amount of resources. So this does not look at a resource gradient. It just looks at how many resources are locally, where am I, and based on some probability from that information, you might stay stationary or you might switch into the moving state. These are increasing and decreasing functions of resources. So if I'm stationary and there are no resources around, I'm very likely to start moving. And that's pretty much it. On this space-time lattice, this essentially boils down to a probabilistic or stochastic cellular automaton. For any students or anyone who's not familiar with the cellular automaton, one basic example is the game of life, Conway's game of life. You may have seen this black and white squares, and they turn on and off based on some simple rules. And so basically that's what this model is. So let me show you a quick simulation. In the bottom panel, I'm just looking at the density of locusts. That's this orange curve. And the density of resources, that's the dotted green curve. In the top panel, I've taken a point for each locust, red, stationary, blue, moving, and I've spread them out so that they all have their own lanes. So these locusts are not interacting. They are just interacting via the resources. So that's so this upper panel is really just a visualization purposes.
Sorry, what's red, what's blue?
Blue are moving locusts and red locusts are stationary. The colors, and depending how big your monitor is, it might be a little difficult to see that. But so you can get a kind of a vague sense of, it's really, I think in this case, something like 10% of the locusts that are moving at any given time. So very quickly, what we observe in this model is that the shape evolves and then reaches some profile and basically has small stochastic oscillations around that profile. But this profile you're seeing at the end is very consistent. And that's this traveling wave behavior. I don't know if you can see. Oh, all right. So the traveling wave behavior is characterized by these two elements. One, essentially constant speed after a brief time of evolution. So here you can see our average speed of the center of mass of the band. And then second is this typical profile that evolves and basically remains stationary up to some stochastic variation, just jumping around a little bit. So a couple of things I want to point out here. It's asymmetric, just like the data we were looking at to begin with. We quantify that with the skewness. It has a peak, which we can measure. Also, in order to really measure things about this profile, we take many iterations and then average. We can measure its width via a cutoff or a standard deviation. And then lastly, so there are these different shape parameters. Then there is also the initial resources, which I'm going to call R+ from now on. And the remaining resources, here it looks like the locusts have essentially eaten everything. Okay, so I introduced this model. I didn't show you very many of the underlying equations, and I certainly didn't tell you about all the different parameters in it. Luckily, because some of these things have been studied before in the biology community, we're able to know some of, and all the control and locus management literature. We're able to approximate some of these many parameters. But we're stuck with about half, 5 of these parameters. And instead of understanding these individual mechanisms, what we have are these global qualities. So there are these 3 shape parameters, the maximum, threshold, etc., or threshold width, etc. And then there's also the speed of the band and how much resources it leaves behind. And what we'd like to do is we'd like to know if our model can reproduce actual accurate observations from the field, because we have those. At first, just in the form of these global observable quantities. So with agent-based modeling, we immediately run into some computational difficulties, and we're not interested in throwing our simple model on a supercomputer. So we'd like to do something a little bit more elegant. And so in order to sample these parameters on the left, we take advantage of the fact that these are collective qualities, and we're going to introduce a PDE model to do that. So the idea will be to take these collective observables and back out individual parameters from these collective observables. So we derived a advection reaction model, which is analogous to our agent-based model. Quick aside, I'm not in the business of proving correspondence here, but we do study that numerically. So the R equation, the important thing here is resources are just decreasing. They're not regrowing on the timescales we're interested in. S becomes the stationary locusts. M becomes the density of the moving locusts. And we essentially have just a transport equation in the bottom. And these terms with opposite signs correspond to a conservation of mass in the swarm of the locust swarm. So again, timescales, we're looking at a matter of hours or a day. Locusts are not hatching or dying during that time. So the total mass n, the number of locusts, is now the total mass. Stationary is in orange or red, and blue is moving. And again, you see it's just sort of a 10% or so are the moving locusts. And you immediately see this traveling wave profile develop in our new model. So next, we take the tools of PDEs and traveling waves, and we hit this until we prove a theorem about that lets us predict these different collective observables. So in the top, I have a very small mass of locusts. In the middle, I've got kind of intermediate, and then a very large mass in the bottom. And you can see that the speed, so they ran for the same amount of time. This one didn't make it very far. This one made it the furthest. So we can predict the speed and also the amount of resources left over. Here, some visible amount, and here some very, very tiny amount on the scale of the plot. We can predict those values. So this theorem, stated more carefully, corresponds to an existence and uniqueness proof for a traveling wave solution to our PDE. And in fact, there are these explicit integral formulas that tell us what the observable outputs will be based on some given set of parameters, including total mass and initial resources. So we end up with this kind of existence curve, which tells us each point on this existence curve, there's a unique traveling wave, which corresponds to that set of parameters. So we'd like to take this PDE theory and apply it to our agent-based model. Here I've just plotted a couple of examples after I think this is 10 million times, or no, 1 million time steps or so, maybe 2, for the agent-based model. And I've centered everything on the center of mass in each image. So visually, our eyeball test, these roughly correspond. And as I mentioned, we did some more rigorous numerical testing of this as well. One other thing about this image is I've plotted 2 images for the PDE and 2 for the agent-based model. In the top image, you can see slow eating. The locusts only eat a little bit. And in the bottom, that lambda parameter is increasing. So they are faster eaters. So this lambda parameter is kind of one of the next parameters that we're going to study. Here you can see individual foraging rate is the lambda parameter. And it turns out that it has one of the biggest effects on the output of the model. OK, so basically, instead of running our agent-based model and sampling this 10-dimensional parameter space and running the agent-based model for long time periods and averaging, we can just solve some simple integral equations, and get these observable outputs. And so computationally speaking, this allows us to sample many more points just using like a standard desktop computer, rather than pushing our rudimentary agent-based model to some really powerful setup computer, supercomputer. So we sample all of these 4.4 million points. We use that to identify which input parameters the outputs are most sensitive to. And then by slicing our parameter space along, here you can see we've got lambda or log lambda on the horizontal axis. So we're slicing along the most sensitive parameters. We iterate that process, and by using information from the collective observables, determine a plausible region of parameter space that outputs observed quantities. And here my little red dot, I think, is the parameters that I've been showing you so far in the talk. OK. So here again is our ABM with optimal parameters. And this is the data from Jerome Buell in Australia. So what I want to highlight right now is that we did not match the exponential tail in our distribution to the exponential tail in his distribution. But when you compare the output of our model based on fitting parameters from other field observations, we end up with a quantitatively good match. So to tie up, for this first modeling project, where we're interested in modeling how this dense front shows up, we're able to leverage the relation to individuals from the agent-based model and the PDE theory to conclude that we've identified a mechanism which really should be included if anybody is interested in modeling these dense fronts of locusts, or interested in understanding how the dense front is going to propagate across a field. So with that said, I'm going to use my last about 15 minutes, I think, to talk about a different model, which is truly two-dimensional. So this model is looking at those columnar structures. We're interested in understanding how these form from this flocking type of collective motion or flocking type of agent-based model. So the elements in here are this pause and go motion, the orientation, and the attraction and repulsion. So again, I'll lay out the model for you. The state variables are just that we have position of each locust and orientation, and then each locust has a state, again, either stationary or moving. Locust switch with a probability, which is no longer dependent on resources. There are no resources left. Instead, this probability depends on nearby neighbors. Specifically, neighbors who are directly in front or directly behind. And most of the action happens in this equation for theta. So each locust has a theta I, and we have an ODE, which describes how its orientation evolves. So I'm going to leave out some of these pieces. I'm going to talk about alignment and attraction and repulsion. So for alignment, remember I mentioned my background is in dynamical systems, and we use this Kuramoto type coupling, meaning I took this function sine in front of the difference here for a locust, a focal locust, and the other locust nearby. And then we weight that by distance. So within this green circle, this locust is aligning its direction with its neighbors through some kind of weighted average, and then the evolution of this ODE. We also tell the locust which way to turn based on who is nearby. So if a nearby locust is in the green region, then our focal locust will turn its orientation to get closer to this orange locust. If the locust is in the red region, it will turn its orientation to get away from the nearby locust. So I haven't laid out all these functional forms and these many parameters, but basically we end up with this set of equations of motion, and we simulate this with a particle in cell method, which leverages parallel computing if you set it up to do so. And more importantly, the thing is, you are not comparing every locust to every other locust. So this is not an order n squared simulation anymore. You only compare them to those which are in that finite range of attraction or repulsion. So this 4,000 locusts runs on my MacBook, my laptop, basically in real time. And you can scale it up with a better, heavier machinery and using that parallel computing. The other thing I want to point out is there were a bunch of effects and even more parameters than our last model. So if we'd like to know whether or not we've really achieved kind of the kernel or the essence of these columns, we need to examine those a little bit. And I think I'm going to, we're going to do so first qualitatively. So I'm running another simulation where I just took out the pause and go motion. And I'm just want to show you qualitatively that it looks here like pause and go motion was not important for the formation of columns, right? And we can do this with each of our effects. And we can ask, OK, did our model reproduce columns or not reproduce columns? But let me jump to the punchline. If we do this without attraction, we get no columns. So to understand this, we're going to again introduce a continuum theory. So hopefully, OK, now the video is over. We still have no columns, and on the left in the full model, or the model with attraction, we do. So here's a continuous version of that agent-based model. So again, we've got state densities instead of individuals, and the density of stationary locusts S and M. You can see these first two equations are very much a two-dimensional version of the equations we used in the 1D case. But now these KSM, KMS, these are the switching rates. These are not depending on resources anymore because there are no resources, right? So it was my dog, Riley. And then we've got an evolution for a theta S, which represents the angle of all the locusts that are stopped at a certain location, and theta M for moving. And so while I wrote it to look like a PDE in the top, this is in fact an integral PDE because of these omegas. All those sums turn into integrations with kernels depending on the distance. So bunch of symbols and equations here, more so than any of my slides before. But what I want to point out here is that if you take S and M both to be constant, and you take the direction of motion to be 0, that is just moving to the right, then that's a steady state of this PDE. And one way to think about this is simple symmetry. If you have a bunch of locusts, they're all marching in some lattice formation, say, the pull to move left and the pull to move right will cancel. So we've got a constant density for S, constant density for M. That's a steady state solution of this integral PDE. And we can do a transverse stability analysis, which basically says, I'm going to put in this small perturbation, and so here's our stationary state S and M, and then 0 for the direction of motion. I'm going to put in this small perturbation of that state, which is modulated by some vertical wave number mu in the y direction, and has a growth rate lambda. So what you see is on the left, once you compute these vertical wave numbers mu as a function of their growth rate, you see that the steady state is stable if you take out that attraction term of the ODE or the PDE, the agent-based model. I'm sorry, in the integral PDE, the continuous model. If you do include the attraction term, then you get that you find that there is a finite band of unstable wave numbers, meaning that basically this spacing is determined by where this curve is positive. This curve represents the places where the growth rate lambda is positive, and those wave numbers will grow in time from a small perturbation. So we have this continuous theory which predicts the qualitative behavior that we were seeking to reproduce, and it ties it specifically to those attraction terms in our model. So that's what I would say is a qualitative or phenomenological result from the PDE theory. But one thing that I've more recently been interested in is diving really deeply into these social interactions. So I'm going to spend my last couple minutes. I do have like maybe 3-5 more minutes. Is that right? Alice?
Yep, sorry. I gave a
Cool.
thumbs up, but you didn't see it.
Oh, yeah. It's right. Gotcha. So this summer, I made a foray into something that was completely new to me, which is motion tracking. So in the left, in the top, you're seeing a video of locusts marching across bare dirt in the field in the bush of southern Australia. So we have been in touch with this ecologist, Jerome Buell, and he graciously provided some old video footage that he has not had the chance to analyze. So this is the project that Alice actually mentioned earlier. I worked with these 3 students in the data science REU to try to pull trajectory data out of these videos. And right. So this is these are our preliminary results at the moment. But basically, we look at two different types of data. One is just the position of each locust in each frame. And that gives us this really nice radial symmetry and a certain length scale for the repulsion and the attractive terms. So this is a heat map showing nearest neighbors with a focal locust who's facing upwards. So you can see it's pretty much radially symmetric. And there's this dark blue area right around the focal individual saying locusts aren't bumping into each other. And this hints to us that repulsion is really a close range mechanism meant for collision avoidance. From the linked track data, we can draw out all kinds of inferred information like orientation and movement speed. And in this, I'm just plotting a radial distribution of the locusts orientation based on a velocity calculated from frame to frame. So you can again see that they're all moving to the left. My locusts have switched direction a couple of times through this talk, but in this video, all these little white dots, which represent locusts in the original video, are all moving basically to the left. And so this bears out in our preliminary data. This preliminary data basically is our proof of concept from the summer, and one of the students is continuing to work with me on his senior thesis, but our hope is to start looking at correlations between, for instance, stopping, so correlations between stopping and nearby neighbors. So for instance, let's look at, we'll use this trajectory data to look at when all the locusts go from moving to stationary, look at each of those time steps and look at who's nearby, right? So we can pull out this nearest neighbor information for particular movement states and look for certain correlations which might hint to, okay, yeah, this locust is stopping because it doesn't want to run into its neighbor or something like this. So this is still very much ongoing work. So just to wrap up, the first model lived in this picture on the left. And we saw that food plays an essential role in the development of dense fronts. Our second model with the social interaction induces columns, qualitatively similar to those observed in the field. And then lastly, in ongoing work, we're trying to get down to those real individual level mechanisms by doing some motion tracking. And I want to conclude with this last image here because the two images at the top are on either side of what I think of as a spectrum. And eventually I'm very interested in posing this as a front stability problem or bifurcation problem, because as you can see, you can have the same species of locust in the same field of vegetation and get these planar fronts and these fingering columns out. So I love this picture because it really hints at how complicated the world is in mathematical biology. That's all I have for you today. So I guess I'd like to thank my collaborators on this first paper and also the various students who I've had the pleasure to work with at Harvey Mudd. Thank you for coming, and let me answer some of your questions.
Thanks, Jasper. If anyone wants to unmute themselves and clap, or you can do the little clap reaction. Yeah, thank you. Hey. And then, yeah, if anyone has any questions, there's plenty of time for questions. And then we'll head over to gather.
So may I ask a couple of questions about the first half? The first question is about the direction of motion. Just because the front is moving in the direction normally to itself, it doesn't mean that every particle constituting this front, every locust is moving in that direction as well. Have you seen any examples where the directions of locusts were not the same as the directions of the front? That's in experimental data. And then, oh, okay.
Go ahead. Well, which way would you rather ask them all up front, or?
Let me ask them one at a time. Sorry.
Okay. So, whoops. Okay, some of my after slides, my collaborators. So in a sense, this is an example of that, right? These locusts don't all move to the left all the time, but overall they do. So in some sense, our assumption that they all move in one direction is an assumption about an average. But taking that, reproducing fronts in vegetation is another active area that I'm working on. And I guess I should be clear. So usually when I say we, I'm talking about some subset of my collaborators from this first paper.
Uh-oh, I think, Jasper, we lost you. Or is it just on my end? Perhaps it's on my end.
Nope.
Okay.
Interesting.
Uh-oh.
If we still have Jasper's co-authors on the call, maybe they can take some questions. Oh, there he is.
I think I'm back, but I don't know if you can see my screen or not, or what you're seeing anymore.
We
You're
do
just
see
seeing
your
my
screen.
desktop?
We see your slides.
You do? Okay. Do you see my thank you slide right now? Great. So how much of my answer came through? Did you hear? Or I was just saying that in some sense, this assumption that they're moving to the left is an averaged assumption. And
so I was not referring to the random variations. Even if you assume that everything is deterministic, if everyone is moving in concert in a slanted way, not norm. So if this is the interface.
Oh, I see what you're saying. Yeah. So what if the front is not perpendicular to the direction of motion?
Correct.
Yeah. So typically in these front propagation problems, so let me go back to one of the really nice pictures at the beginning. Here. So there is definitely some curvature to this front, right? And you might even say that this area is like the flattest area, but I think in the end, my answer to your question is there have not been specific observations where individuals move in a direction that is not perpendicular. But in some settings, that front is curved. So still perpendicular to the local curving front. Am I closer to what you're asking now?
Yes, thank you.
Yeah.
I'll wait with the second question until others had a chance to ask something.
Could I ask a question?
Yes, please.
Yeah, that was a really cool talk. I guess I'm just trying to make sense of putting the two different parts together. Since the waveform you're showing here, like this huge front, not columns, is formed by locusts that are gregarious and so presumably attracted to each other. So I guess now I'm wondering why they don't always form columns. Or are there some conditions where the attraction doesn't result in the formation of columns?
Yeah, I think those are exactly the kinds of big questions that we'd like to eventually answer in the end. Let me go back to this slide, and I probably need to share my screen again. Let's see, this one. So you can see my messy desktop. Okay, so certain species of locusts do have a propensity to to form certain collective shapes or patterns, right? The Australian plague locust is observed to do both, kind of both ends of the spectrum, but often in areas of lush vegetation, they'll form these fronts. Desert locusts are observed to have dense fronts, but much more often form these columns, and sometimes it's some combination where you have a column forming, but in the front it's denser, and you can examine an exponential tail. So the question of, I mean, one possibility is maybe that, let's see, maybe the locust's interaction with resources is strong enough for the Australian plague locust that when they're in those environments, that resource attraction, for instance, overcomes their attraction to individuals, which would normally form columns. And I'm interested in formulating that in a mathematical context where, for instance, you can do some stability analysis that basically says as you change this one parameter, this front bifurcates into a column or fingering instability. Yeah.
Thank you.
I'd like to try a question.
Yeah.
So yeah, it could also be that like the behavior is governed by the sparsity or lushness of the vegetation. So have you thought about that? I mean, inhomogeneous vegetation basically looks like one big difference in Australia and the desert.
Yeah. Yeah. I think that, I mean, there's also interesting questions about formation. So, you know, let's say that these locusts form in some field and then start to migrate across area of desert where the vegetation is inhomogeneous or non-existent. And how long will the dense front persist before kind of being replaced by these columns and this more constant density? We haven't, one of our, I have one simulation. I didn't put the video in this talk. I don't think it's even in these slides, but basically it's, what if you take a column and you run it into a field of vegetation? And how does the form of that locust, that simulation, how do they stop or change? Yeah, I'm gonna scroll to the bottom of my hidden slides and just see if I have that simulation in here, because that would be a great one for your. Yeah, I don't have it. I'm sorry.
That's okay. I'm sure that you've been experimenting with numerical results you're not ready to show yet, because you could put resources in those 2D models and see what happens.
Exactly. Yep.
But you don't have the story yet, I'm sure. Thank you.
Yeah. Any other questions? Or Alex, if you wanted to go take a second turn.
Sure. Thanks. So in this first half, you're using your numerical experiments primarily with Pdes because they are cheaper computationally to calibrate the model parameters. But presumably those model parameters have evolutionary nature in that they have evolved to maximize some utility function for the locus. Have you looked at the result of your simulations to figure out the individual locust experience and what they're getting out of different parameter values and why the ones which you're seeing are better for them than the others?
Yeah, so we were sort of talking about this earlier today, right? A little bit. So we had along with Andy, who I can see now is in the audience, we had a student who kind of just started to get us into that area this summer. But we don't have any results. We're not even sure how exactly we want to formulate that question at this point. but the student basically said, took our 1D model and ran it over many generations. And that was, so, and he was asking, you know, in which setting do most locusts get to eat the most, or something like this. Yeah. You feel free to chime in, Andy.
Yeah, I mean, the question that a lot of evolutionary biologists are looking at is that this gregarization behavior among locusts has evolved something like 7 independent times on multiple continents. So there's got to be an evolutionary advantage. And there are qualitative and semi-quantitative models that basically look at this in terms of food foraging. And, you know, this is somehow optimizing the species surviving when resources are sparse. But that's sort of the state of the art. So we'd love to be able to get to the point of tuning parameters, but that seems a long way off at the moment.
Yeah, thanks for reminding me that I left that out at the beginning. We talked about different species of locusts on different continents, but it is some small evolutionary miracle, or perhaps just the design of the system, that that this behavior has evolved apparently independently.
Any other questions for Jasper? Okay, thanks again, Jasper. If anyone here wants to join us in the gather space, we'll head there shortly. The link, Erica posted the link in the chat.
And thank you all for coming.
Thank you for the talk.